a merit award from NIAID. It's the Methods for Evaluating Vaccine Efficacy, and it was originally started as an R01 20 years ago and was recently converted to a merit award. And with that, I do statistical and epidemiologic methods for evaluating vaccines, and I'm moving into surrogates of protection, which is what I'm going to talk about today. I'm also, because Hulin mentioned, and Martin, other networks being represented at this meeting. I'm a PI of one of the research groups of the NIGMS-supported MIDAS network. That's for modeling infectious diseases, and it includes a lot of, in that we do both differential equations, we have large stochastic simulation models of populations, and we're also quite interested in estimating transmission parameters and statistically. And we're starting to build in genetic data into that. And so Hulin mentioned the forward paradigm and also the inverse paradigm. And so these grants represent both forward and inverse paradigm. And Ira Longini and I have been working together for over 22 years now in both those paradigms. I'd like to thank Peter Gilbert, whom some of you know from the HIV work, for some of the slides that I'll present today. And I'd also like to mention the Fourth Summer Institute on Statistics and Modeling of Infectious Diseases that I'm the director of and founder of at the Department of Biostatistics at the University of Washington. And this is the website. And here we teach 13 different courses. The courses are 2.5 days long, so you don't have to commit to coming out for 2.5 weeks. I don't have any scholarship money I have money from NIGMS to support this, so I have scholarship money for students and postdocs. And this year, I just looked at the web, there are 153 people enrolled to take an average of 2.5 courses each. And the other thing that occurred to me when I was preparing the slide is that I have 13 courses, but I actually have room for 15 courses. I have money for next year, and I'd like to have a little bit more in computational biology or rational vaccine design. If anybody thinks of a course that they think they'd love to teach, the pay scale is not medical but statistical, but it's a lot of fun. So if anybody wants to teach a course, just talk to me and we can see about offering something new next year. Okay, so to the talk. So this is an outline of the talk, and I'm going to start out just briefly on concepts of definitions of immune correlates and surrogate endpoints, and introduce two different paradigms, the predictive and the mechanistic. And a lot of what we've been talking about in this particular meeting has been the mechanistic version and causative version of surrogates. And my focus is going to be on predictive approach. It's a different approach that some of you may have heard of or may not, but I hope it'll be food for thought. So we'll talk about, first of all, Tier 1 correlates of risk, Tier 2, which are surrogates of protection, specific surrogates of protection, and my focus will be hopefully to convey what the difference between a statistical and a principal surrogate is, and then briefly about the Tier 3 general surrogates of protection, and briefly to conclusions. So what exactly is an immune correlate? And this is a relevant conversation. I'm working on a PO1 on dengue right now, and Monday I spent most of the day trying to define what a correlate of protection is with the people I was working with. And but the concepts are quite surprisingly elusive and often misunderstood. And the methods for assessing the validity of surrogate endpoints are really quite subtle. And I'm going to present just one small aspect of that today, and hope we'll get a point across. So just for those of you who don't work in vaccine efficacy, which is my area, usually in a randomized study, at least, if you're lucky enough to have a randomized study, you would have a vaccine arm and a placebo arm, and then each arm would receive inoculations, and the placebo would receive either a placebo or control. And then in the vaccine arm, you would measure the immune response. And you'll notice here in this particular diagram, it doesn't mention measuring the immune response in the placebo arm, but you might you'd probably measure that too, but that's part of what we're going to talk about. And then you follow them up in the general phase 3 trial. You look at a clinical endpoint, either infection or disease, or depending on what disease you're looking at. And so the primary objective is to measure, evaluate the vaccine efficacy to prevent pathogen-specific disease or infection. And a secondary objective is to assess the vaccine-induced immune responses as correlates of protection. So the importance of immune correlate. So finding an immune correlate is the central goal of vaccine research, and it's also one of the grand challenges, global health from NIH and also the Gates Foundation, and we've had some nice talks here on HIV and TB, and there was a poster on malaria. And sometimes I would say it's a holy grail, but people pointed out that's maybe not politically correct these days. But the immune correlates are useful for, if you could find a good immune correlate, you can shorten the trials and reduce the cost. These are phase 3 trials are extremely expensive. It can also guide iterative development of vaccines between the basic and clinical research, because you might be able to improve your vaccine if you know what's actually producing the good response to the good protective response. It's useful in guiding regulatory decisions, and some vaccines have actually been licensed only on the immunogenicity data, and a recent example of that is the Meningococcal A vaccine that was licensed for Africa, the Menafric vac was licensed just on immunogenicity alone, and of course then one has to go in and try to correlate what it was licensed on with the actual disease. And it can help in guiding immunization policy, and in boldface, you can also bridge efficacy of a vaccine observed in a trial to a new setting, and that's what people really want to do, and that's the really hard part, and I'm not actually going to talk about that a lot because it goes beyond the time. So a lot of the talks here have talked about different aspects of immunity to different infections, but it's actually the correlates for licensed vaccines. A lot of them were licensed on the fact that they prevented disease, but without really understanding the immunity. And so here we have 3 different categories of vaccines by whether little is known or nothing about what the correlates are, the intermediate and the high. And you can see that there are a lot of vaccines that are quite effective that people don't really know well why they protect. So what exactly is an immune correlate? So there are 2 distinct ways of thinking about an immune correlate, and one is the prediction paradigm and one is the causal mechanism paradigm. And as I said, the focus on this talk is going to be on the prediction paradigm and define these 3 different levels of immune correlates and summarize the statistical frameworks for their assessment. But there's been some back and forth recently in the literature between Plotkin and also some people out at the Hutch, between the causal, and Plotkin's a big man in vaccines, the causal agent paradigm, and this would be if you have a marker that is mechanistically causes the vaccine efficacy against a clinical endpoint. Whereas in the prediction paradigm, you'd have a marker that reliably predicts the level of vaccine efficacy against the clinical endpoint. Now, a lot of the discussion in this symposium has been on trying to find things that actually predict the immunity, or looking at the immunity and the mechanisms of the immunity, but this is more of a statistical framework. And both are useful for vaccine development, but they're assessed using different approaches. I mean, what I'm going to talk about is a little bit different than what everybody else has been talking about here. And so the goal of statistical assessment for surrogate endpoint validity in an efficacy trial, we use the prediction paradigm. Okay, but thinking about surrogate as a mechanism of protection, again, you need a lot of approaches outside of the trial, just like what had been discussed in the last 2 days. You need the basic science, you have to understand the specificity and the functionality, you have to understand all the effects of vaccination, you have to understand the disease process, the pathogenesis, the laboratory validation studies are necessary. Again, we've had some discussion about that. And we've also had here at the symposium causal manipulation of animals, which I'm not going to comment on, little mice sitting there getting all, but that's also part of understanding how the infection works. But then, okay, so let's go to people where we really care where it's going to work. And so I'm going to talk about this hierarchy of immune correlates that was produced by some of the people out at the Hutch and published. It's been published in a lot of different papers now. But this is the one thing I would like to get across, that this is a hierarchy of immune correlates definitions, and it was published in a quite readable paper back in 2007. And it's been published and discussed in lots less readable papers since then in the statistics literature. But the first level would be called a correlative risk. That's what's called Tier 1. The second level is a specific surrogate of protection, and the third one is the general surrogate of protection. And the correlative risk, the brief definition is the biomarker correlates with the clinical endpoint measuring vaccine efficacy. That is, if you have an antibody and you look at the rate of, I'll go into this in more detail in further slides. And you can measure that maybe in vaccine efficacy trials or epidemiologic studies. They don't have to be like randomized studies. A specific surrogate of protection would look at the vaccine effects of the biomarker and what it predicts in vaccine efficacy for the same setting as the efficacy trial. And for that, you need like a large efficacy trial or multiple similar trials. And for the general surrogate of protection, it would be something, and that's what we're ultimately really interested in for the long term, is it would be a specific surrogate of protection that predicts vaccine efficacy in different settings. That is, if you change vaccine lots or vaccine formulations, or you look at the populations in Africa versus Asia, or you have different viral populations, that this is a surrogate you can really depend on to come through in a lot of different ways. And evaluating something like that is really quite difficult. And so the point is that this is a hierarchy in that a general surrogate of protection is also a specific surrogate of protection, and a specific surrogate of protection is a correlative risk. So I'm going to go into a little detail about this now. So Tier 1, correlative risk. So the definition of a correlative risk is quite simple. It's an immunologic measurement that predicts or statistically correlates with the rate of the clinical endpoint used to measure vaccine efficacy in some population. So that's a quite easy thing to look at. For example, individuals with higher antibody titers should have a lower rate of pathogen-specific disease, or they not should, but it could be. That would be a correlation. You run a regression and it correlates. And you can get that in an observational study or in phase 3 trials. And here's an example of looking at a correlate of risk. So this is from the VaxGen phase 3 trial that many of you are familiar with. I was on the DSMB of this trial, which was an interesting experience. But the VaxGen trial, they looked at antibody response for different correlates of risk for HIV infection in the vaccine group. I think it's interesting, given how bad the vaccine was, that so many publications have come out of the analysis of this study. But if we look at this, so what you see here in this graph is the 4 different quintiles by CD4 blocking level that they measured in the blood, so Q1, Q2, Q3, and Q4. And the relative risk, we're looking just now in the vaccine group, and it was looking at antibody data measured on infected and uninfected vaccinees only. So we're assuming that the first quantile of CD4 blocking level is the comparison, so we have a relative risk of 1. And what you can see here then is that at Q2, Q3, and Q4, you have a relative risk that goes down a little bit, 0.4, 0.3. So that's compared to relative. So what it looks like here is that you have a correlative risk with the antibody level. Another example is an old trial of flu vaccine, and we've had some nice talks on flu vaccine here. So this was 1943 trial that was done in Michigan. They had a big operation those days. 1776 men, that's an interesting number. And they looked at a trivalent vaccine that contained the Weiss strain A and the PR8 strain A. And they were assigned more or less randomly, alphabetically ordering of names. They didn't really do a lot of randomization back in those days. And they measured the strain-specific antibody 2 weeks post-inoculation in every 10th vaccinee and every 5th placebo. And the clinical endpoint was the hospitalization due to respiratory illness with specific influenza strain found in throat culture. So, and they had, as you can see, a short follow-up time just at the end of 1943. And I'm going to focus on the results for strain A in this particular talk. So what you can see here is that, this is another example, you have this placebo group at the top and the vaccine group at the bottom, and the antibody titer on the x-axis and the percent in the group that had that antibody titer. And so what you can see is that the antibody titer in the placebo group was in general lower than the antibody titer in the vaccine group. And what we can also think about is it's influenza, and so we know you get repeated exposures to influenza over your life. So unlike many infectious diseases, the placebo group would actually possibly have an antibody titer that you could measure. And we'll talk about the problem with other diseases soon. And what you can also see here is that the correlation of risk, so here we have plotted for the placebo group and the vaccine group, the case incidence by log antibody titer. And so what you can see in this graph, I suppose, the problem with these things is people sort of start doing things like that. So I don't like to use a pointer if I don't have to. In the placebo group and the vaccine group, you can see that the case incidence goes down with the log antibody titer, so that we do see in both the vaccine and the placebo group a relationship. But, okay, so now we have a problem. So a correlate does not a surrogate make. I'm sure many of you know Tom Fleming. And there are reasons why correlates of risk fail to be surrogates. I'm just going to list them here without going into detail because I want to go on to another point in this talk. But they can happen in many ways, as you can imagine. The biomarker, or whatever you're looking at, an antibody, is not in the pathway of the intervention effect. That is, the antibody and the vaccine might have completely different pathways, and so it's insensitive to the effects of the vaccine. The biomarker is not in the causal pathway of the disease process. It may correlate, but it really doesn't is not really a surrogate. And the intervention has mechanisms of action independent of the disease process. That is, the vaccine might do something that is not really relevant to the disease process. So anyway, we'll go on to tier 2, the specific surrogates protection, and this is going to be sort of the focus for most of the rest of the talk. And we consider evaluation of a candidate surrogate based on a single efficacy trial. Okay, so the definition here of what we call a specific surrogate of protection, or specific SOP, is an immunologic measurement such that vaccine effects on the marker reliably predict VE, the VE is vaccine efficacy, for the same setting as the trial. So that's specific. That is, you know, if you moved to Africa from Asia, it might not work, but we're just, you know, keeping it within the trial. So, for example, the subgroups with no vaccine-induced antibody response have no efficacy, and subgroups with large vaccine-induced antibody response have large vaccine efficacy. Now, already at this point, given how complicated some of the talks have been and the kind of complex immunological reactions that we've been looking at in networks, we could replace at some point the antibody response with network or some other aspect of the immune response. It doesn't, this is for this talk, just what we're talking about. Okay, so now there's 2 different definitions of the specific surrogate protection. And if I can get one thing across, is I hope that understanding a little bit the difference between a statistical and a principal SOP is what I can hope to do today at the end of on Friday. So the statistical SOP is based on an approach defined by Ross Prentice back in 1989, which is quite famous in the surrogate literature, not just in vaccines, and it's based on observed associations. And the principal SOP, surrogate protection, it builds on a paper by Frangakis and Rubin, who are statisticians here on the East Coast, and their definition of a surrogate endpoint. And this is based on what's called the potential outcomes framework for causal inference. And I don't know how many of you know anything about causal inference. It's not for the faint-hearted, but there is a course in the Summer Institute for it if you're interested in learning more about it. That's an area I work in myself. Okay, so the statistical SOP, statistical surrogate protection, satisfies the 3 criteria set up by Prentice. That means it would be vaccination is associated with the immunologic marker. We would expect that, of course. The immune marker is a correlative risk in each of the vaccine and placebo groups. This is what we sort of looked at in that graph about influenza. And the relationship between the immune marker and the clinical endpoint is the same in the vaccine and placebo groups. And that's a tough one. That is, after accounting for the marker, vaccine and placebo assignment doesn't have any information about the clinical risk. So if you imagine that if I measured your antibody titer to something, that I wouldn't have to know whether you're vaccinated or not. I would know how efficacious what the correlation is between that vaccine and that's a tough one. So the interpretation is of this final one is all the vaccine effect on the clinical endpoint is mediated through the marker. Now that's a tough one. We'll see how tough it is actually for things besides flu. Okay, so we'll go back to this example of the 1943 influenza vaccine field trial. I'm sure you've heard of some of these people like Salk and Francis. I forgot to mention that. So the incidence of hospitalization with the Y strain A in the placebo group was 75 of 888, and the vaccinees 20 of 888. So that gives a 8% versus 2%. So the vaccine efficacy, for those of you who don't know, is 1 minus relative risk. So the vaccine efficacy estimated here is 73% based on the clinical outcome. And so the goal is now, for the next few slides, is to assess the Y strain A antibody titer as a statistical SOP based on the Prentice criteria. Okay, so we already saw this graph that in the vaccine group that the antibody titer is higher than in the placebo group, so criteria 1 holds. Criterion 2 is, we look here, we see the Y, we already looked at and we saw the Y strain A antibody titers are inversely correlated with the case rate in each study group. We already saw a version of this graph. This graph contains both the observed and expected based on a logistic regression. The green is placebo and the blue is vaccine. That looks very similar. And then, so this is basically the same graph again. We can also check the consistency between the correlative risk models in the vaccine and placebo group, and it looks very similar in the two groups. And as a matter of fact, you can do something further than that if you believe in regression modeling, logistic regression. You can stick both the antibody titer and the vaccine assignment into a logistic regression model. And here we have, we want to focus on model 3, which is in the dark blue box. And so we have the intercept, the log titer, and the treatment. In this case, it's vaccine. And what we see is that the log titer is almost -1 with a standard error of 0.12. Often we'd want to do confidence intervals, but we can do standard errors here. In the vaccine, which is TRT, is 0.33 with a standard error of 0.32, so 2 times that would be, so you can see that you go through 0 basically on that particular intercept, that particular thing, so that you can see that the vaccine assignment does not affect the influenza risk after controlling for the log antibody titer. So in this case, it's a nice example, and criterion 3 holds. I'm not going to go through the PR8. It doesn't hold in that case, but time doesn't allow me to go through that. But the other thing you can do is you can do a further validation of the statistical SOP, and you can do the estimated vaccine efficacy versus the predicted, and you do the predicted by looking at based on the correlative risk model for the placebo group and the distribution of the antibody titers in the vaccinated. It's just a little transformation. And you get 73% in one and 82% in the other, and that's okay. It really doesn't work for the PR8. It's a nice example, but we don't have time for that. Okay, so that's all very nice. And it works very nicely for that example, but there are some problems with the statistical surrogate protection approach. Okay, so the one that we kind of already alluded to is that the immunologic measurement is a response to pathogen-specific protein or proteins. And you would expect in most cases that the trial participants have never been infected with the pathogen. And so the immune response would be 0 or non-response in most cases. And in fact, you sometimes screen people beforehand and say, we will only take them if they're immune naive. And so then you have a problem that in the placebo arm, that you can't look at the correlative risk model in the placebo group because inside criterion 2 can't be evaluated because it's 0. So there is no correlate of risk. And also it's difficult to check criterion 3 for full mediation because you can't really look at what the effect is because you have no variation in that group. And so it's not really that easy to even do a statistical surrogate protection approach. But there's another more basic approach, okay? So first of all, checking the full mediation entails checking for each immune response level. This is like the next 5 slides, if I can get this across, and I'll be very happy. So equal clinical risk between the groups, okay? We need to actually look for each immune response level, the vaccines with marker level at S versus placebos with marker level S. So we're going to call S now as the level of whatever marker we're looking at. But S is measured after randomization. And therefore, this comparison may reflect selection bias, potentially misleading about the marker's value as a surrogate of protection. And this is a limitation pointed out by Frangakis and Rubin in 2002 in a general context. They weren't talking about vaccines, but it's been picked up a lot in the last 10 years in the vaccine world. Okay, so this is an illustration of how this works. So imagine that we have a binary immunologic measurement. So it could be positive or negative, it doesn't matter. And consider an unmeasured covariate reflecting the strength of the immune system. So we're not measuring it. So there's some latent variable in their strong, weak immune system. And suppose we have a randomized trial of 4,000 people, and we randomize 2,000 to vaccine and 2,000 to placebo. And so by randomization, we would expect the distribution of this unmeasured covariate to be equal in both groups. I mean, it's a big, 2,000 is a lot, so we're going to say that it's the same in both those groups. And we're going to say that it's 50-50 so that in each group, 1,000 have weak immune systems and 1,000 have strong immune systems. Okay, so now you go and you vaccinate. You vaccinate, of course, only in the vaccine arm. You don't vaccinate in the placebo arm. So you vaccinate in the vaccine arm, and the ones in the 1,000 people who have the weak immune systems, only 10% have a positive response. And we're going 0, 1. And it's actually easier a little bit on a continuous scale, but we'll say only 10% respond to the vaccine in the weak group, and 90% in the strong group. Now, what we mean by weak and strong might be that if they get infected, they have worse outcome. They get sicker, they have a higher viral load, they have, their weak immune system, so they have a shorter time till they develop AIDS or whatever disease you're thinking about. And so now, what is the problem here? That is, we have, if we look at sort of in this pie chart, so now if we're going to look at a statistical surrogate of protection, and remember, we didn't measure, we don't know who has the weak and strong immune systems. That's an unmeasured confounder. So we're comparing apples with oranges, because now suppose that the people who have the weak immune systems and they have, they didn't respond as well to the vaccine for some reason that we can't measure, and if they get infected, they have this more severe outcome. That if you can see here, we have 90% of the vaccine group has weak, who might get infected, 90% of them will have the worst outcome. Whereby in the placebo group, it's 50-50 still, so the people who get infected there, it's going to be 50-50 weak and strong, as opposed to 9. So what you could easily conclude from this is that your vaccine makes people worse off, when all it is a selection bias that you haven't been able to pick up because you weren't able to, you don't know who has the weak and the strong immune systems. So that this is what the source of this selection bias potentially is for if you're looking at a statistical surrogate protection, because you're measuring some measure that is post-randomization. So the alternative is this approach is called the principal surrogate framework. And this leverages augmented data from vaccine efficacy trials. And this is sort of the idea that's new, and you might be able to, some people may have heard it, but when the first time I heard it, I thought I was a little crazy, but then it was a long time ago already, so I'm used to it now. So, and if we go back again to the example I presented before, where we looked at the CD4 blocking level as a correlate of risk in the vaccine group, and we were just looking in the vaccine group, and you've already seen this graph. So here we have the 4 different quartiles, and we have the relative risk we set this relative risk at 1 in quantile 1, but the problem is here, so it looks here. So what are we actually looking at here? Let's think about this a little bit more. We can't really tell, looking at this, whether the fact that these people had the low response to the CD4 blocking level, the ones with the low ones, that might have been just that they have a low response because they're the ones with the weak immune systems, and they're somehow more prone to get infected Not that they're more exposed, because we wouldn't expect that to be related to this, but it could be that once they get exposed that they're more prone to infection because they have the weak immune systems, and they also have the low response. Or we could also come to the conclusion that the vaccine, if you just have a low response to the vaccine, that it actually enhances your probability of getting infected. We can't distinguish that on these data. So that's a problem, because of course if the vaccine is really at that level causing you to get infected, I mean, it's making it easier for you to be infected if you're exposed. That's a really bad thing. So now we could change the comparison and now compare it to the placebo group. So the only difference here is that the relative risk of one, the scale's been altered here, so it goes from 0 to 3.5, so it's a different scale. So now the placebo group is considered the comparison group, and so now you can see that the quantile 1 of the vaccine group actually has a higher relative risk than the placebo group, whereby the other two go down a little bit. So the relative risk of infection in quantile 1 of this group is about 0.75. I know what the answer is. So it's 1.75, where the relative risk goes down in the higher ones. And so now we're thinking, how does this change things? Well, that makes it look a little different, because now we're seeing that it's worse off than the placebo group. However, there's still something wrong with this, because the placebo group includes all 4 quantiles. So we've grouped here. We haven't broken the placebo group out into its different quantiles yet. Okay, quartiles. I keep saying quantiles, but it's quartiles. Anyway, so now, okay, so this is where we take off the ground a little bit. Okay, so what does this mean? So now this is how do we want to assess the immune response as a principal surrogate of protection? We have to be able to know what the response is in the placebo group. And of course, the question is here we have the different quartiles, and we can look at the rate of infection in the vaccine group by quartiles, because we vaccinated them and we measured their response, but the placebo group didn't get vaccinated, and presumably they don't have a response because it's not flu. Okay, so then here's some fruit: apples, oranges, bananas, and pears. And so what we want to estimate by the value of S, we want to be able to estimate vaccine efficacy by quartile. But of course, there's some question marks here, because we don't know what the rate of infection is in the placebo group in that quartile. So this is a hypothetical example 1, where we could imagine, and this is just an imagination, we could imagine that we know that in quartile 1 in the placebo group, it would also be 0.18 and 0.18 in vaccine, and the same in 0.10 and 0.10 in quartile 2 and 3, just same as vaccine, and in quartile 4 would be 0.08, and in both it would be 0.11 in both groups without being broke down, and then by quartile the vaccine efficacy would be 0 everywhere. And this would mean that this particular value, this antibody is not a principal surrogate of protection, because VE doesn't change with the value of the surrogate, with the antibody. Now, if we could imagine another example, again, hypothetical, where in the placebo group, that in fact, we would know that if we had vaccinated them, and so they would be in quartile 1. In fact, we didn't vaccinate them, and so they have 0.11 all across the board in quartile 1, quartile 2, 3, and 4. And so in quartile 1, we can estimate vaccine efficacy under these conditions to be minus 0.64. It's negative. That means it does make it worse. And then at 0.09, 0.09, and 0.27. And so we get a vaccine efficacy that increases with antibody titer, ignoring the fact that it actually makes things worse in this example. But that's what we're talking about when we wanted to be able to estimate vaccine efficacy based on the immune response the placebo group would have had, but they don't actually, if they had been vaccinated, but they don't get vaccinated. So the question is, how can you know that? Okay, so this is an equation. So this is a heuristic definition then. So we define the vaccine efficacy where S is the vaccine efficacy as a function of the immune marker is 1 minus the risk of infection for vaccinees with antibody S to vaccine over the risk of infection for placebos who would have had antibody titer S to vaccine had they been vaccinated. That's this potential outcome framework. And so where you can think about hypotheticals. And so the interpretation is the percent reduction in clinical risk for groups of vaccinees with antibody titer compared to if they had not been vaccinated. Okay, so then the definition of a principal surrogate is an immune measurement satisfying that the vaccine efficacy in somebody who has a measurement of 0 of the surrogate would be 0. And I had to leave this fancy word in here because it shows up on the next slide. And this is what's called causal necessity. I won't go into that in any more detail. And you also would really like to have some large variability. That is, you'd really like to have vaccine efficacy change with the value of S. And so here's a graph expressing that. And here we see on the x-axis the immunologic measurement S, whatever that might be. And the vaccine efficacy on the y-axis is a function of S, and this only goes down to 0. It's only from 0 to 1. And the immune measurement goes from 0 to 1, but we could imagine it goes negative. But what we see here, this black marker is just flat. So the black line, it's the black line is flat. And so it doesn't change. Whatever, if a marker would be like that it doesn't change and your vaccine efficacy doesn't go up, it would give you no information about vaccine efficacy. Now, the blue and the green markers satisfy what we call this average causal necessity. That is, at 0, if the measurement of the S is 0, the vaccine efficacy is 0. And as it goes up, they both go up, but as we notice, blue goes up a lot better than the green does. And so there's a lot of variability here in this one. Now, the red one doesn't satisfy this idea. The red one doesn't go down to 0. So it doesn't satisfy this condition that the vaccine efficacy has to be 0 if the marker is 0. And so that red one wouldn't satisfy that. Now, why is this of interest to us? Well, something that's been discussed here in several of the talks, and I'm sorry I can't sort of rattle them all off right now, but several people talked about being interested in vaccine efficacy and improving the vaccine target, that if you can find a marker that actually has a graph like this blue one, where if you can produce with a vaccine, there's a marker that if you have a high level, this is where the red circles are, if you have a high level of that marker, you have a high efficacy, then you can say, well, I want my vaccine to produce something that makes a higher level of that particular marker. And you can do an iterative process. That's where you can go back to the clinical side and say, to the research side and say, I want my vaccine. And Tom mentioned that, that Barton Haynes now is in one of our discussions, Barton Haynes is heavily involved with the HIV work, and he's heavily influenced by these thinking. And so they're targeting, they have found things that they think are surrogates, and now they're trying to really hone in on those B cells or whatever, the different cells. So that's really been, so that's what the conclusion there is that there's a difference between a surrogate principle and a statistical surrogate of protection and a principal surrogate of protection. And it's conceptually the same as evaluating treatment efficacy in subgroups. So you can think of these different baseline values of S as a subgroup that happens, but you'd prefer it to happen before the randomization. So principal stratification makes S equivalent to baseline covariate. Okay, so just very briefly then to finish off in a couple of slides. So there is a challenge to evaluating the principal SOP, the principal surrogate protection, that there is something wrong with this. Okay, we don't know what the immune responses to vaccine are missing. They're missing for the placebos. That's sort of the weak link in all of this. Sounds nice, but Dean Folman, who's the head of the biostatistics branch at NIAID, and came in from Heart, Lung, and Blood, or Blood and Lung, whatever, I can never remember. But anyway, he came in with a different background and brought in some different ideas. So he wasn't afraid to suggest something new, and said, well, why don't we just do something wild, like fill in the missing data somehow, and we could measure a predictor of the immune response in both vaccines and placebos, or we could do a closeout placebo vaccination. That is, when the trial's over and the people who didn't get vaccinated, we'll just vaccinate them and see what their responses were. And there's a large literature in this that Peter Gilbert has been promoting and doing a lot of. So this is a schematic of this idea. I think the idea is pretty clear. But so the top is the vaccine group and the bottom is the placebo group. And so obviously in the red is the vax and the W. You could imagine, okay, before we vaccinate anybody, let's just give them all hepatitis B vaccine and see what the responses are in the placebo and in the vaccine group. We're just going to vaccinate them with something else and see, and then we're going to see how that correlates. I'll go into more detail, but in one more slide. But anyway, so this is what we call the baseline predictor approach. So you find something that might correlate with the response to vaccine, and you measure it also in the placebos. It could be a lot of other things. Okay, and I'll show you how that works. Or you wait here until the end of the trial. You can see here where it says SC. So at the end, when for the people who haven't been, and the red vax there, so when the trial's over, you go in and vaccinate the people who didn't get infected. Now, there are some problems with this approach as well, but that's the general idea, that you try to fill in what's happening in the placebo group. And so this is just a brief picture. So here at the top, we have the vaccine group, where we've measured, say, the response to hepatitis B, and S, which is the S that we're really interested in the vaccine group. And the bottom is a control group, and there's a W that we've measured on the placebo people. And based on that, we'll impute their S. And so based on that, then the other one is, I've already gone over this, you don't need to do that. And so just as a sort of a fun example, Peter took the information from that flu trial that I presented and made the assumption, and there have been some talks at this meeting that assumption isn't totally crazy, that assumed that in the antibody observed in the placebo group, that it was inversely related to the response to the vaccine, and they imputed the vaccine response. So here you see the antibody observed in the placebos and the imputed as the antibody that they would have had to the vaccine, and then they did the computation based on that. So they estimated the vaccine efficacy based on the S as 1 minus the case rate for vaccinees with antibody titer S compared to the case rate for placebos who had the imputed antibody titer S. And they got a nice response. So what you see here is on the x-axis the antibody titer S to vaccine, and on the y-axis is the estimated vaccine efficacy based on that S. So then for each antibody titer level, you can see that the vaccine efficacy actually goes up as the titer goes up. So that was just okay. So this is where the vaccine development may be improved by this prospective research to discover and measure baseline predictors. And it's kind of an odd idea. In the flu trials, we could look at the baseline predictor. You might be able to get the antibody or some other measures, but there aren't very many infectious diseases like that. And one of the things that they are doing now is they're gearing up to do a new phase 1 HIV vaccine trial in South Africa. Part of that trial is actually to vaccinate both the placebo and the vaccine groups with hepatitis B and tetanus. And they're going to look at, as part of the outcome of the phase 1 trial, what the relationship is between the hepatitis B and tetanus vaccination to the HIV response. And they're going to see about whether they might use this approach to look at surrogates in the actual phase 3 trial. So this is affecting how. So that's basically all I have to say about that. I have one slide only on general SOP, which is what a lot of people are really interested in, is that's being able to take some measure of immune response and use it to move vaccines to new populations, or if you've got back-to-back, you have a new vaccine, a new formulation, you can't really do back-to-back efficacy trials, and they're just way too big. And so what you want to be able to do is predict this, and you need to do meta-analysis, and actually it's not clear that anything works. I think that's the bottom line. It's a very difficult topic. And sort of a final slide. There's a new paper that just came out this month that I don't have time to go into, but it's by Plotkin and Gilbert in Clinical Infectious Diseases. It's a very short paper and very readable that if you're interested in this topic, I can highly recommend, because they talk about how to reconcile these different nomenclatures without getting into anything technical. And so they talk about correlative protection and divide it into mechanistic correlative protection and non-mechanistic correlative protection. And basically, then you can think of it as here the surrogate endpoint. The correlative protection is the predictor of efficacy, and the mechanistic surrogate would actually have, be a causal pathway. It'd be many of the things we discussed at this meeting. And you could also have a non-mechanistic surrogate, and I don't have time to go into that. But one example that they give is the Meningococcal Vaccine, where a mechanistic surrogate is the bactericidal antibodies. And one knows that really has how that works in making the vaccine efficacious. But you can also, if you vaccinate, you also get a good ELISA response, but it's not clear that that has anything really to do with the efficacy of the vaccine. So that's an example where the mechanistic, and these are, the mechanistic and non-mechanistic approaches are, they're exclusive and they're exhaustive and mutually exclusive. Okay, so my conclusions. Okay, so we've had a lot of talk here about modern data like microarrays, proteomics, microbiomics, and so the problems are similar, but as you all and we all make more advances in understanding more about the infectious disease process and the immune process, we're going to be able to use these approaches to also be surrogates, but evaluating these surrogates in actual humans is going to be very similar. And they also may yield earlier predictive surrogates, aiding the surrogate assessment and improving their practical ability. So basically, bottom line, what this meeting has brought with all those big teams that everybody puts up, it's all highly interdisciplinary these days. And we don't need to read through all of this. So Tom Fleming is a very wise man, if you've ever met him. Surrogate endpoint assessment is one of the most important problems and one of the most difficult. And on that note, I'll leave you and say thank you.
It seems not a question.
No question. Oh, there's a question. Oh, no. Hi. Yes.
So I was worried about the problem of what happens if you have two surrogates. Because let's say you have A and B, and they both actually have some interesting effects. And let's say you don't know about A. Then your requirement that when A is 0, the vaccine has no effect, you won't meet that requirement because in fact, if either A or B can correlate with some protection. As long as you're high in one or the other, you have good protection. Then what you'll do is reject that surrogate, even though it may be an extremely powerful measure of prediction. So how do you deal with things when you suspect, which I'm sure is often true, that there's more than one?
Well, yeah, I understand. So the question is, what do you do if there's more than one? And I think that's a really good question. Obviously, this is a very simplified talk. And that is a problem. I mean, can you put them all into a model? I mean, a lot of this is a science. I mean, it's not just this, but there are, I think one of the challenges brought up at this meeting is, what if you have a whole network and you have to have that whole network operating? And so it's no longer an antibody, but a network. And that whole thing has to sort of gotten mobilized. And I think that's a good scientific problem that's not solved in this setting. I will just say there is some literature also, and I mean the statistical surrogate. It said it was supposed to, that's a statistical surrogate, but there's, they've expanded this whole principal surrogate of protection to the point of looking at the strength of prediction. How much of the outcome is predicted by a particular antibody response or by a particular thing. So it's gotten much more complicated than.
But I think there may be an extra issue there that some of the simple situations may be, for example, a toxin and an antibody, where the protection may be by a single mechanism, and then these definitions work very well. But with some other vaccinations where, as you pointed out on one of the early slides, we don't know a lot about the actual mechanisms of protection. If they, in fact, they're multiple, they're not a single thing, not just an antibody binding, then there may not be a good single measure that you can use that would meet these definitions.
That's true. I mean, what would you do in that case?
I'm not sure. Your guess is better.
Students, take note. What would you do in that case? Now, that's a good point, and obviously, yeah, I can't give you a really good answer right now.
But it may mean that you would be a little more accepting of the measures that don't drop down to zero, where the crown is zero and the efficacy is zero. If you lessen the requirement for that to drop to zero.
Right. No, I agree. That's
As long as you've got a reasonable slope.
as long as you've got a reasonable slope. Right. I'll have to give that a little more thought, but I think that's a good point. I mean, obviously, yes, I think that's a very good point. Thank you. So
Possibly along those lines, possibly not. I'm thinking that everything that you said about surrogates would also apply to linear combinations of surrogates.
right, you could model. Yeah, that's
So
right.
there's no qualifications necessary to take what we talked about today to linear combinations, right? Everything translates into
Right, you could do linear combinations, you could do complicated combinations, you could do network. I agree with that. You could put several things into your model. I'm worried about this particular point right now with the dengue, and people are, I'm worried about this particular point with the dengue. I'm thinking about a dengue thing, and they're measuring all sorts of things, and I'm thinking, and they thought, well, we'll just measure all these things, and you can just model it and tell us what the correlates of protection are, And I'm thinking, well, I don't think it works quite that way. You have to think about what you're doing, but I'm also very skeptical about like multiple regression models, and you put a lot of things into a model, but you could do linear combinations.
Okay. And again, I'm thinking you want to follow up more on something like that. Is there any changes that need to be made for therapeutic vaccines as opposed to prophylactic vaccines?
For therapeutic vaccines.
Right. You said translated as well for therapeutic vaccines.
I don't work in therapeutic vaccines, but I think of therapeutic vaccines more as treatments. And it might work. One area I have worked in, as I said, I work in causal inference, and I think a lot about post-infection selection bias and the people who, and so it's a slightly different, to answer a slightly different question, there's a problem in post-infection selection. If you randomize and people get infected and you're interested in post-infection outcomes, then, this is a slightly different answer to your question. If you're interested in, say, like if people get vaccinated and then they get infected and you're looking at HIV, and you're interested in whether people who get infected have a worse, does the vaccine increase your viral load or decrease your viral load? If you look at the people who get infected and follow them, they are no longer a random sample, because people who get infected in the vaccine group, as I pointed out, are not necessarily comparable to the ones in the placebo group. However, if you were looking at and this came up in a discussion recently, it's again not quite the answer to your question. If you were looking at, if for example, in that trial where most of you probably read about it, it came out with big to-do, where they were looking at discordant partners and they gave early treatment to the HIV-infected partners to see if it prevented transmission. In that one, randomization occurred after the people were infected. It didn't occur before they were infected. So the people who got randomized were properly randomized. They were infected. You weren't looking at people who were randomized when they were not infected. Then they got infected. They were no longer a random sample. And so I guess, in your case, if you're treating people, the treatment, the vaccines, I consider them treatment. You would randomize after the people were infected. And so it's a little different problem, because you would randomize them after they got infected. And so you have a proper comparison groups. And so it's a different problem. It's a much different problem with treatment than it is with prophylaxis. It's a little different problem. I don't know.
So, last question.
Last question.
Yeah, just trying to understand one thing with Dean Bowman's suggestion to vaccinate at the end stage of an HIV trial, to vaccinate the placebo group and see how they respond. Since you're only vaccinating people who are in the placebos who are HIV negative,
Right.
Again, have selection bias, because the ones that are HIV negative might have stronger immune systems or something, and will respond to the
Somebody was
vaccine.
gonna ask that.
beforehand.
Thank you, Alan. No, I actually have exactly the same problem with that. I mean, I've never really understood how he can get away with that. But he is the head of the biostatistics branch, so. But I always have this, I was preparing the talk and I thought somebody's going to catch that one. So, but I agree with you that there's something a little fishy with that approach.
Okay, so if we have more questions,